Okay, good dear students. Today we are going to study and learn about a special technique which is very often used in microbiology, and the technique is a microscopic method which is known as hanging drop method. As the name itself indicates, it is a method where we hang the drop on the surface of a cover slip, and by using a specialized slide, this cover slip is placed in such a position that this specialized slide, which is called a cavity slide, the drop will hang. So the appearance is like a drop hanging from some surface. For example, if we put a drop on a surface of some metal or a plate at the bottom of the plate and we lift it, so what we see is a drop formed at the bottom, and this drop is in a hanging position. Now that hanging position of a drop is placed in another cavity or a slide. These things will become more clear when we see the diagram and we study the technique. Now this is a type of wet mount. What is wet mount? By using microscopes, we observe organisms which are not easily observed by unaided eyes. And to observe them, we use various staining techniques. When we use the staining techniques, the organisms which are being stained are dead at the end of the procedure. We prepare a smear, stain them, and we put a drop of immersion oil and observe. That is a normal staining methods. Now the wet mount means it involves the observation of organism either in a water as a mounting medium or as some other material where we observe them in a natural state without staining them. However, it requires an expertization for observation and an eye to look at the organisms under unstained conditions. Why it is difficult? Because microorganisms are normally colorless. The light passing through the microscope is colorless. The lenses are colorless, and therefore there is no contrast observed. So you have to create a contrast by adjusting the amount of light entering the diaphragm and condenser. So these techniques are what we call a wet mount. Wet mount is basically a drop of medium which contains the organisms to be observed. And this is placed on a microscope and it is directly observed under lenses. As I have told you, since they are colorless, the type of technique which we use is dark illumination in order to see the colorless organisms. And since we are observing the organisms under objectives in natural conditions, you can find the organisms which are living and which are moving also. So, this hanging drop method is widely used for one important thing is to observe the motility of bacteria, to check whether the bacteria are motile or not. Do they have that ability to move from one place to another? So, hanging drop preparation helps to see bacteria alive in their environment. This technique was invented by Robert Koch in 1878. Remember, Robert Koch was one of the first bacteriologists. He, along with his contemporaries, that is D. Pasteur, established the science of microbiology. So he was the first one to develop this particular technique. Now, why known as hanging drop method? This is why it is known as hanging drop method. See the diagram. This is a cover slip which I have lined it in red. This is a cover slip. And the cover slip is having, or we place a drop of bacteria in a liquid in the center of the cover slip. But this is the final position you will be observing. How it is placed, we will see it in a stepwise manner. So the drop which is being placed is like this. Now, why it is in a hanging position? It remains in the hanging position throughout the experiment because the slide, this is the slide, microscopic slide. And this microscopic slide, as you can see, it is a thick slide and it has got a depression in the center. This is the depression and this is the slide. So in the center, there is a depression, or we call it as a cavity or concavity. It is a concave in nature. And this cover slip is having on its four corners Vaseline or petroleum jelly. So it seals the cover slip. At the same time, cover slip being attached to the slide, but the drop still remains in a hanging position. And this position, it gives a very good view of microorganisms under natural state. What are the advantages of this method? It preserves the cellular shape and the arrangement. It is not distorted at all, unlike normal staining techniques. It prevents the drying out because we have applied Vaseline, we have applied petroleum jelly, and therefore the drop remains there in a place for a long time. It is not evaporated fast. The disadvantage is the method can be risky. Why? Because we are observing the microorganisms under living natural conditions. And therefore, if pathogenic organisms are present, it leads to the exposure of a technician or a person observing the slide. And therefore, it is risky. Now, what are the materials which are required for this process or the technique? First, glass slide. It is a normal glass slide with depression. Depression means cavity. And therefore, this slide is also known by name cavity slide or concavity slide. Now, surrounding this, you can apply adhesives, tape or paraffin ring. So what you require is a paraffin ring. You require wire loop for taking a suspension. Cover slip is required, which is to be placed on the cavity. Then for observation, you require microscope. You require Bunsen burners, because between burners we will take up, with the help of a sterile wire loop, we'll take a drop of or a suspension drop of a culture. And for the beginners, we usually use a culture of Proteus mirabilis. Proteus mirabilis, basically, it is a non-pathogenic organism. In few cases, it may be involved in urinary tract infections, but it is an opportunist pathogen, to be exact. Otherwise, it is non-pathogenic. Now, these are the materials which are required. Cover glasses, that is the cover slip. Depression slides, that is the cavity slides. Toothpicks, or you can say wire loop, and petroleum jelly. Now, first step, slide preparation. How the slide is being prepared? We take a clean glass slide and apply a paraffin ring or an adhesive tape. There is a, suppose this is a cavity which is in a center. Then surrounding this, you apply here the adhesive tape or a paraffin ring, paraffin or a Vaseline or a grease. So apply, put few drops surrounding this cavity slide. Always remember to handle the slide from its edges. Do not use your fingers on the slide or on a cover slip. Then place a loofful of fresh culture broth. Which organism? Proteus mirabilis or Proteus vulgaris, which is to be tested or which is to be observed in the center of a cover slip. And we are not using a heavy inoculum, it is just a light inoculum. And it should not be very turbid. Remember, why? The explanation for that I will give you at a later stage. Then this suspension is placed in the center of a cover slip. The cover slip is placed on a flat surface, and the glass slide, that is the cavity slide, which was lined with, or the cavity which surrounding that cavity, which was placed with some grease, the slide is inverted on a cover slip. It is placed over a drop so that Vaseline will seal the cover slip around the concavity. Next, the slide is turned up, upside down turning is done, so that the cover slip comes on the top and it allows the organisms to settle, or it allows the drop in a hanging state on the cover slip. Allow it to settle for a minute and then observe in a microscope. So this is the diagram that is there. This is a cover slip. In the center of the cover slip, with the help of a wire loop, we put a drop of suspension of Proteus vulgaris or Proteus mirabilis. This is a cavity slide. This is a slide. This is a cavity, and surrounding there we have a petroleum jelly. So this slide is inverted and placed on a cover slip. After placing the slide, it is turned upside down. So ultimately, this is the position of the slide which is observed. Say this is the one. Grease is applied on cover slip. It can be applied on cavity, periphery of the cavity, or it can be applied on the cover slip. Second step, a drop is placed on the center. This is the drop in the center. Next is we are inverting. This is the slide which we call it as a cavity slide, and it is inverted. Inverted means the cavity comes at the bottom and then this is pressed on the cover slip. So when this is pressed on this cover slip, the drop which was placed here will come here. It is still on the cover slip, and finally when it is firmly pressed, this cover slip will be sealed by grease. Finally what is done is it is inverted. The entire combination is inverted. So the cover slip comes on the top, the drop is hanging in a cavity of that special slide. So this is how it is observed. Clear? Now, next step is to examine under microscope. When we examine under microscope, first it is examined under low power. Under low power, you have to first adjust the slide in a such a way that the edge of the drop is observed. This is the edge of the drop. It means you can imagine that this is a drop, this portion is a drop, and this is the other outer portion. So you can we can imagine that the drop will be like this here, or you can say it will be like this. Drop is here. Okay, and this is a little portion of the drop that we are observing in a field. In an objective field, only we observe a little portion of the edge of the drop. So with the help of adjustments, you have to shift the slide in such a way that you clearly observe the edge of the drop. So this is the edge of the drop that you can see here. So this is the edge of the drop. Okay. Now, you have to focus here. Now, when you focus the edge of the drop, always note that here the microscope needs certain spatial adjustment. What kind of adjustment? As I have told you, the water is colorless, the microorganisms are colorless, the light, visible light is colorless, lenses are colorless, and therefore everything being colorless, there will not be any contrast observed. So it will be very difficult to observe. And hence, in order to visualize, you have to create a contrast without use of dyes. And this can be achieved by allowing or moving the condenser down. Under oil immersion lens, we have to ensure maximum light, but here the light required will be less. Light entering the condenser should be less. So what we do is, the entire assembly of the condenser and diaphragm is switched or it is moved down. The diaphragm is also partly closed. I'm not saying fully closed. It is partly closed. So that the amount of light, because of these two adjustments, that is the condenser moved down and diaphragm partly closed, the amount of light entering the microscope will be quite less. This would create a contrast. Okay. After the edge of the drop is clearly observed, without moving the stage, we switch on from 10x objective to 45x objective. That is the high power objective. From a low power objective, it is switched over to high power objective. And when it is switched over to high power objective, again this is the drop. As you can see, this will be the drop here, like this. Entire drop will be here. And this is the edge which you had mounted under low power. So this will be clearly observed here, the edge of the drop, and you will see the organisms here. These are the organisms. This is the center of the drop. This is the edge of the drop. And on the edge, you will see more number of organisms. So this is the observation under 45x, that is high power. Again, when you use high power, you will have to increase the amount of light as compared to low power. So you may need to move the condenser slightly up and diaphragm slightly open. This is in comparison to what we adjusted the microscope under low power. As compared to low power, condenser should be slightly more up and the diaphragm should be slightly more open, but definitely less than the 100x, that is normal staining procedures, which is observed under oil immersion tanks. Now, question arises, why to mount the edge of the drop? Why we observe the edge of the drop? There are two reasons for it. First reason, the drop will have a different refractive index as compared to plain glass, and therefore it can be easily searched. Second, the drop contained the microorganisms, and these microorganisms, specifically aerobic organisms like Proteus vulgaris or Proteus mirabilis, they are aerobic organisms. It means they require oxygen for their activity. They respire oxygen. Now the amount of oxygen is definitely more at the edge as compared to the center of the drop. Say for example, in a big lake, if you are in the center of the lake, the chances of drowning are more because you will be immersed, you will be drowned in the water, and you will not be able to stand erect because it is having a high depth, the amount of oxygen is less. So what you do if you are drowning, if you are there in the center, you will try to swim towards the edge of the lake. Same thing the microorganisms do. Microorganisms, they swim towards the edge because at the edge the depth is less, more amount of dissolved oxygen is there, and therefore they will all be moving towards the higher oxygen concentration. Oxygen gradient is less at the center. more at the edge, more at the periphery. You can call it edge or the periphery of the drop. And therefore, all organisms, they are moving towards the edge. And when you observe, you will be able to see the microorganisms moving in a random motion. This motion should not be confused with the Brownian movement. What is Brownian movement? It is an auxiliary, it is a movement like a pendulum, oscillating movement, where due to the bombardment of the molecules, water molecules, the molecules will keep on moving. It will force the microorganism to move at a constant speed. But this oscillating movement is to and fro movement. It means the movement is like this, from here to here, always like moving like this in a particular fixed area. While random motion, which is due to true bacterial flagellar movement, it is like this. Suppose this is an organism, then this will move from here to here. This is the kind of motion which will be observed. This kind of motion, which is by microorganism, is due to the normal flagella. While Brownian movement, which was discovered by Brown, Dr. Brown, it is due to the bombardment of water molecule. The water molecules, because of the collision between themselves, they force the bacterial cells to move from their respective position. They compel the bacteria to move in an auxiliary fashions, oscillating fashions, pendulum-like movement. It is not a random movement. So here what you have to search is these dots which you are observing in the diagram. Each dot represents one bacteria. These are all different bacteria, thousands and millions of them. So this kind of movement is known as normal flagellar movement. Okay. One point which I would like to mention here is, once you prepare a slide, you should observe the slide as early as possible. Because as I have told you, all organisms present in that drop would like to move towards the edge. And it takes only few minutes, 2 to 3 minutes maximum, by which time all organisms will be moving towards the edge. Once they come to the edge, they settle there. And then there will be a lot of crowd. You may not be able to see them very clearly, or you may be able to see the Brownian movement, which may be interpreted as a flagellar movement. And hence, this observation of a hanging drop preparation has to be done from a fresh broth, which is a young broth, because the young culture will have a better motility, better flagellar movement as compared to the older cells. Hence, take care to prepare the slide and observe it as fast as possible. So this experiment, hanging drop preparation, is an experiment to check the motility of the organism in question, whether the organism is motile or not. This is one of the most confirmatory tests for checking the bacterial motility. So this is about the hinging drop preparation. Okay, thank you very much.